Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really fascinating guest today involved in creating a better tomorrow. We have the honor today of being joined by Dr. Amy Proal, who currently serves as the President and Chief Executive Officer of PolyBio Research Foundation, as well as the Chief Scientific Officer of the Long COVID Research Initiative. In her work with both the Foundation and the Long COVID Research Initiative, Dr. Proal is involved in conceptualizing and coordinating various large-scale collaborative research projects among research teams studying infection-associated chronic conditions such as long COVID, as well as other conditions like long Lyme, chronic fatigue syndrome. Dr. Proal has written multiple review articles over the years that focus on the core biologic drivers of long COVID and some of these other conditions. She's a regular speaker at various infection-associated chronic disease conferences like Lyme Mind, International Congress on Autoimmunity. Dr. Proal holds her bachelor's in science and biology from Georgetown University. She did her Ph.D. in microbiology from Murdoch University in Australia, and her work has been focusing on examining these molecular mechanisms by which a broad range of microbes, as well as fungi, viruses, serve to dysregulate human gene expression, immunity, and metabolism, and ultimately how the dysbiosis of the human microbiome or the virome contributes to chronic inflammatory disease processes. As a member of the research team of the Autoimmunity Research Foundation, she's authored papers and written book chapters for organizations like the Ventnor Institute, the Autoimmunity Network, and she's lectured at NIH and numerous other U.S. and international conferences. We're honored to have her with us today. Dr. Amy Proal, welcome to our show.
Thanks for having me on.
It's great having you. I would really like to start off, as we typically do, by sort of handing you the floor for a little bit. You have, beyond the amazing background I read about you, you have a very interesting story behind why you got involved in this whole area in the first place in terms of microbiology and dysbiosis of the microbiome. Talk a little bit about yourself, if you would, and a little bit of your personal health experiences and why you got so interested in this space.
Sure. So when I was a pre-med student at Georgetown and doing really well, actually great, just acing the classes and doing fine when I got an Epstein-Barr virus infection. And I got really sick. I just basically tanked and then became almost bedridden from the symptoms that I got after that. And I tried to figure out what was wrong, but when I went to most of the doctors that I saw, they really had few answers. And in fact, some actually just said what might be a psychosomatic issue. In other words, that maybe I was just making it up, which is not great, but all right. So it soon dawned on me that I might have to figure out what happened to me on my own. So largely from my bed, I began to read a lot of papers on infection. And the reason I did that was because my symptoms started after an infection, and also because when I was young, so I have a fraternal twin sister, and when between the ages of 3 and 5, and I don't know why, I was vaccinated for measles, mumps, rubella, but I got, when I was very young, measles and rubella on 2 separate occasions, and I was hospitalized for them. They were really severe cases. I almost died each time. It was a big drama. I got a separate case of meningitis that I was also hospitalized for. I got a case of pneumonia that was very severe that I was hospitalized for. I was also hospitalized for a case of scarlet fever. It was a little ridiculous, honestly. And then I was okay. And my twin sister didn't get any of those infections, interestingly. And then after that, I really was okay for a long time. When I was in middle school and high school, I was captain of the tennis team. I was okay until I got this Epstein-Barr virus infection and then got really sick. So I was interested not just in that infection, but I was interested in the possibility that cumulative infections or different infectious hits might be able to add up in some way to contribute to a chronic disease process. And that perhaps some of the pathogens that I got in the infections when I was young might have not fully left my body. Maybe they were still there in low amounts or in low levels. And maybe the Epstein-Barr virus infection sort of, affected my immune system in some negative way that allowed them to sort of emerge from what might have been latency or dormancy and begin to drive more symptoms. So to better understand that, I started reading a huge amount of literature, like I said, on infection. And it turns out that in the 1960s and 70s, there was really a robust research community on persistent organisms in chronic disease. And there were teams the one tough thing about that time is the technologies that teams could use to study organisms. For example, they would get human tissue samples and other, samples and look for organisms that might still be persistent in a patient. Was that the tools they had to find those organisms were somewhat limited. So at that time, they were using, kind of a classic scene from a laboratory, Petri dishes, where they would really, if they were going to find the organism, need to culture it, which is a very specific process that requires a very specific medium. We now know that a lot of organisms that persist in the human body don't grow in culture, so that there were great limits to what they could find. But even then, they reported a lot of different bacterial viral pathogens in samples, tissue samples, sometimes blood samples from patients with chronic inflammatory conditions that seemed really similar to mine. And even in conditions like Alzheimer's disease or diseases deemed to be later autoimmune, right? And so I found that really interesting. So I reached out to some of those researchers and actually befriended them. And they began to tell me more about their work. They actually sent me some of the textbooks that they had worked on and used for this. So I learned a lot from that and became really interested in infection. And then I began basically to do an experimental treatment that I somewhat helped come up with a team of other people, where the goal of the treatment was pretty straightforward. It was to use a potential drug that can activate important parts of the immune response. And again, more research is needed. This is experimental. We tried to do some research on this, but more research would have to be done for what this drug really does to the immune system to be solidified. But I know other options. So I started to do this, and I also started to take some pulsed antibiotics. I started to take some natural antivirals, some herbal antivirals, and I started to try to treat what might be infection. And I did actually gradually improve by doing this. And I saw, it's a long story. I saw a lot of different people and practitioners as part of this process. And as I started to improve, then I thought, well, look, I don't want to be a doctor anymore, because I don't think doctors even have enough options now on what to do. I want to be a scientist, and I want to study these infections and these pathogens more so that we can better understand exactly what they're doing when they contribute to chronic disease, and then sort of move that into medicine so that it can actually be better applied. So I switched, and that's where I decided to get a Ph.D. in microbiology, and I was able to do that. And then from there, I continued to have an incredible interest in this topic. And last but not least, even when I was getting the microbiology PhD, I was still a bit sick. So it was hard for me to stand up for long periods of time in certain labs. So I was actually allowed to do a PhD that in some instances was more literature-based, and I could piece together parts of what we're working on in different labs. And what that showed me was the value and the necessity of actually connecting work in different labs, because there would be one lab who might not realize that the lab next door to them had the equipment they needed or had samples that they could work on. Everyone's very busy, and there can be a huge lack of communication. So I became interested in that space of how people working on different sort of selected topics can actually combine their knowledge collectively to work on larger research projects that tackle more comprehensive topics. And that's still what I primarily do today with PolyBio Research Foundation.
That's awesome. That's really awesome. As you're talking through your own experiences, I was going through PubMed and looking a lot of what you've been publishing over the years, and um, just last year you on this very topic of sort of these post sequelae disorders. You published in Frontiers of Microbiology in June 2021 this paper, Long COVID or Post Acute Sequelae of COVID-19 or PASC, an overview of the biologic factors that may contribute to persistent symptoms. And in this paper, you dive into so many aspects of this. You talk about obviously the impact of straight from the virus on our tissues. B, the ability of COVID to reactivate, as you're saying, these other viruses that may be hanging around our virome, and there's parts of us from years ago. And then you go into the whole sequelae of inflammatory response, of the immune response, what may be happening there. And then you connect, this is COVID, long COVID is, this is a horrible thing, but hey, we've seen this before in different areas. You talk about chronic fatigue syndrome. You talk about what the people with Ebola have been experienced in terms of this post-Ebola syndrome. Take a little time, if you would, sort of to walk us through this paper, because I think, this coalesces, as you're just saying, bringing together a lot of pieces of this. Talk a little bit about sort of where we've been in terms of these post-sequelae disorders, and then take us a little into long COVID per this paper.
Sure. So that's a paper I wrote with my close colleague, Mike Van Elzeker, who is a Harvard Mass General researcher who also studies ME/CFS and long COVID. ME/CFS is what we call chronic fatigue syndrome, myalgic encephalomyelitis, chronic fatigue syndrome. And Mike and I had been working together for several years before the COVID pandemic started on that condition, ME/CFS. And to give you more context on that condition, it's a condition in which patients often develop very debilitating chronic symptoms after other viral infections. So that's a little bit of where I was diagnosed with that, the Epstein-Barr virus or enteroviruses, which are other viruses that are single-stranded RNA viruses that, similar to SARS-CoV-2, are often acquired via respiratory infection. It can reach the gut sometimes and actually can drive cases that are more symptomatic or asymptomatic. There's a lot of parallels between the enteroviruses and SARS-CoV-2. And those viruses had already been implicated in ME/CFS. For example, there's a team in California that had found those the genetic material and proteins of those enteroviruses in intestinal tissue from patients with ME/CFS. So when the pandemic began, and we unfortunately, Mike and I and several of our colleagues at PolyBio Research Foundation, unfortunately knew that long COVID was going to become a thing. Mm-hmm. And that's because almost every major pathogen, viral, bacterial, even fungal and parasitic in some cases, it depends, has been connected to a chronic syndrome in which a subset of patients who get that pathogen don't fully recover and develop chronic symptoms. So like you mentioned, Ebola there's a condition called post-Ebola syndrome. There's also post-Zika syndrome. There's all these different, syndromes where you have this core common trend in which someone gets an infection, but for some reason, some people recover and some people don't, right? So with long COVID, when the pandemic began, there was a lot of course, people coming into the space and saying, how do we research this? What might be happening? And I was actually, even at the time, contacted by some other journalists and by some other groups to speak. And I was surprised that what I would hear at these other conferences or interviews was that long COVID seemed like a complete mystery, that we would just seem to have to start from scratch and that we had no leads on what might be causing long COVID. It was sort of a narrative at that time. And Mike and I thought, no, we have plenty of leads. We really do. If you actually look at post-Ebola syndrome or some of these other conditions and all the research that's been done on them, you absolutely can come up with exactly what we did, core biological trends that you can at least position as some of the most likely to be, again, occurring in a similar, condition at the end of the day, just with chronic symptoms after a different virus, right? So yeah, we pulled from that literature, and post-Ebola syndrome is an interesting one and is a big clue, because with post-Ebola syndrome, again, we're talking about some people getting chronic symptoms after Ebola. It was studied for a while, and then there were really some actually big breakthroughs with that condition where it turns out that, and this is really true and really important to understand in these chronic, post-infectious conditions, or whatever you want to call them, what we call them infection-associated chronic conditions, is that the virus, if it sticks around in someone and doesn't fully clear over time, it usually isn't easily found in blood anymore, because blood is just not really a smart place to be if you're a virus. That's where the immune system is really robust. There's a lot of immune cells that are going to recognize you. So in simple terms, viruses and even other pathogens will hide in tissue where they're a little bit, can ensconce themselves a little bit more, right? And that's what we call a viral reservoir. That's what we're calling in long COVID, a little bit of virus left there but it can continue to provoke the immune response or drive, gene expression changes that can be detrimental. And in post-Ebola syndrome, it had already been known that, they hadn't been able to find Ebola in blood or some common secretions, but when they looked in eye tissue, they still found the virus, right? When they looked in semen, they were still finding the virus. They became called, in that case, anatomical sanctuaries, but very similar to viral reservoir, right? So to the point where there was actually, around the time when long COVID was becoming more of a, understood condition, that there was actually a new Ebola outbreak that was sparked in Africa. And 4 different teams concluded that the new outbreak was started by someone who must have carried the virus persistently for years at a time in a reservoir. So it's just we knew then that one of the central trends that we could position into long COVID off the bat was the straightforward possibility, and that's one of the first sections of our paper, that maybe at least a subset of patients who are developing chronic symptoms after SARS-CoV-2 may just not have fully cleared the virus from their system. In other words, they no longer are testing positive via a nasal swab test or some kind of easy test like that, but the virus has burrowed more deeply into tissue, into intestinal tissue, into the tissue of their lungs, into a lot of internal tissue sites that are actually really hard to sample and get to. So they might seem fine on blood work, but might still have the virus in those internal sites. So we wrote about that as one of the possibilities. And that continues to be, honestly, the topic in long COVID that continues to emerge as the central topic today. There have now been teams, there's a team at Harvard, David Walt's team, that used a really sensitive technology to look for SARS-CoV-2 proteins or antigens in the blood of patients with long COVID. And they found a high proportion of the long COVID patients in their study still had the spike protein circulating in blood. In their paper, they conclude, and of course they're not sure, but their major hypothesis for that is that the virus likely is still persisting somewhere in tissue in a reservoir, but maybe the spike itself buds off, maybe in an exosome, a little vesicle, and that the spike, this is more common with the proteins that viruses create, the spike might get into the bloodstream, and you can almost measure that as an inference of the fact that the person might still have the virus. So that was a really compelling study done by a really compelling team. And there's also been, another team who looked in intestinal tissue of patients that were also working on a larger long COVID study with now got intestinal tissue from patients 7 months after initial illness and still found in some of that tissue SARS-CoV-2 RNA, SARS-CoV-2 protein, right? So there's even a large NIH autopsy study, literally called SARS-CoV-2 persistence throughout the body and brain, where this was an autopsy study, so tissue examined after death, but they found the RNA and protein of SARS-CoV-2 in a truly wide range of human body sites, just different tissues, nerves, organs, you name it, just still there. So certainly the virus seems capable of persistence in any of these infection-associated chronic conditions. I always try to, there's no need to complicate an illness if you don't have to. And so it seems, and that's part of the research program we built, the Long COVID Research Initiative. That's part of what's a PolyBio initiative on specified on long COVID. The research teams involved in that, our first round of projects center on that, because it's this sort of essential question that you want to know first. Do patients still have the virus or not? It's sort of like question one. And that knowing that, having a lot more data on that then allows you to interpret related data, for example, on clotting or, blood vessel issues or vagus nerve signaling with more context, because you can then understand, hey, maybe it's the spike protein of the virus that the patient still has that might be driving part of the clotting processes, right? So you're able to then understand a lot of the other phenomena just more accurately by having addressed and studied that question first. But to study that, And that's a lot of what we've done with our research program now. You have to get tissue samples from patients. And that's a lot of what we've worked for the last year to build, is we've found doctors who can get biopsy from patients with long COVID via biopsy procedures, who can get intestinal tissue, lung tissue, skeletal muscle tissue, lymph node tissue, these different tissue types from patients very safely, very carefully, right? There's a lot of time that goes into making sure that sample collection is okay. And then those samples are sent to teams that are using some of the top technologies in the world to examine those samples. And the point of using some of these new tools and technologies is to go far beyond the question of this SARS-CoV-2 and its proteins there, but to also be able to capture, and we can do this now, these tools are computer-based actually. We can capture the whole, basically, all of the genetic material from the sample. We can pull it out and then we can map it to even other organisms that might be in the sample, because we know the genetic code of those organisms. So we can sort of use the computer to make that match. And then we can also understand the activity of individual immune cells in that sample. So we can understand, let's say if the virus is there, we can then know if the immune cells near it are acting differently than in a different part of the tissue. And that allows us to say, maybe we now understand what the virus might be doing if it's there, how it's modulating the immune response, how it's changing the immune environment, right? And then we can even sequence and understand how the human genes in that sample are turning themselves on and off. And we can understand if there seems to be a difference in that expression, which we can also correlate with the presence or absence of the virus to understand if it's affecting the way our human genes are working in that sample, because viruses and their proteins do alter the way our human genes can work. They hack our pathways, which is one of the major ways they drive disease. So that is a big focus of what we're working on in this research program, although there's many other components as well. And then going back to the paper that we wrote, I believe the second scenario we talked about is that, which is also just a robust area of study in our research program as well, that you could have SARS-CoV-2 and maybe you still harbor the virus, or maybe you clear the virus. But either way, at some point, the virus knocks down the immune response in a manner that allows other pathogens the patient already harbored, but were in a latent or controlled state, to themselves reactivate and drive chronic symptoms themselves. And SARS-CoV-2 can effectively do that, because one of the things that it does is it creates a number of proteins that knock down the activity of what are called interferons. And interferons are proteins that are part of the immune response that play a central role in controlling viral infection generally. So if SARS-CoV-2 knocks that down and you have, Epstein-Barr virus or a different herpes virus or another, parvovirus, other viruses that people can have that are sort of under control if their immune system is working more robustly, those viruses might be able to reactivate, infect a new tissue site, infect a new nerve, change their activity, express more proteins, and themselves drive a wide range of chronic symptoms. And we know that that's no joke. The herpes viruses, for example, which there is ample evidence now, are reactivating in patients with long COVID. So Akiko Iwasaki, who's part of our initiative, already published a really amazing study. It's a preprint now on long COVID patients. And part of what they did note was increased reactivation of Epstein-Barr virus in their long COVID patients. Of course, not in every patient, it differs, but yes, that's a trend. In Epstein-Barr virus it can drive cancer when it's active. It's been connected now to multiple sclerosis. There were 2 seminal studies recently published at Harvard and Stanford that just really recently pulled Epstein-Barr virus into MS, literally showing that in the MS brain, what we were previously calling an autoantibody is actually an antibody that hits Epstein, tries to respond to an Epstein-Barr virus protein and then cross-reacts with a human protein in the brain. And that kind of cross-reactivity with the Epstein-Barr protein is a huge part of the MS tissue that get targeted in the human brain, right? So we have these viruses, while that reactivate in long COVID, being increasingly tied to all kinds of really actually serious other chronic diseases. So it's not a stretch that is a big issue, and also potential driver of symptoms. And then, after that in the paper, I think where we go next is that you also have to consider a really important consideration, which is that humans are not sterile.
Yeah.
We're actually seated with just trillions of interacting organisms. And we collectively, we refer to those organisms collectively as the human microbiome and human virome. And the microbiome is bacteria, fungi, archaea. The virome is, DNA and RNA viruses, but also bacteriophage.
Mm-hmm.
Bacteriophage are viruses that infect bacteria, and by doing so modulate their activity. So there's this whole ecosystem of organisms that are continually, have their own habitats in our bodies and are themselves interacting. It's a little like a rainforest under conditions of health, where the immune system, again, is more robust and active. What happens is that we have these communities of organisms in our guts and our lungs now, and in a lot of human body sites. There's a growing number of sites that are now understood to harbor these microbial communities. And under conditions of health, it's like I said, in a rainforest, there's animals that can cause problems. I mean, you have animals that can kill people or can cause issues, but because the ecosystem's balanced, they're doing this in a fairly regulated way, right?
Mm-hmm.
Yes, one animal eats another, but then there's a balance to how that happens that keeps the overall ecosystem in okay shape. And that's the same for the human microbiome. But if the immune response goes down, which is something that SARS-CoV-2 can definitely do, or there's just other, stressors on the patient, even the other reactivations of pathogens, that, the immune system is no longer able to sort of, I would say, regulate the organisms in microbiome communities as well. And what's important to understand is a lot of the organisms in these communities are what we call pathobionts. Mm-hmm. And what I mean by that is that they can act as commensal organisms, just basically friendly organisms, if you want to call them that, under conditions of health. But under conditions of imbalance or inflammation, they can change their gene expression. They can change their activity to act as pathogens. Mm-hmm. They can act, they become troublemakers. And an analogy I've used there sometimes is, let's say you had students in a classroom and the teacher was there, right? Well, the teacher's the immune system and the children are the organisms. So when the teacher's there, they're behaving, right? But if the teacher leaves the room, the immune system, those same kids, right? They're going to get up to all kinds of trouble. So that happens. And what we refer to that as dysbiosis. Now that these microbiome ecosystems with all these different organisms are thrown out of balance and other potential pathobionts and pathogens emerge, they can cause secondary infections. It can be really difficult. So what we definitely then are also trying to incorporate into the picture of long COVID is, even if let's say you still harbor the virus or you still have these other issues, your immune system's been depleted, what happens with the microbial communities? And there are already studies that have shown, for example, there's a study that showed the oral microbiome of patients with long COVID is more imbalanced and there's an overgrowth of problematic bacteria. That can be a big problem because oral bacteria can leak into the blood. And then once those organisms get into the blood, they can cause other problems. So there's a lot of flow-on effects when you start to get organisms in these body sites, because the barriers that are sort of supposed to keep them in that community, they can become permeable when the organisms are imbalanced because there's more inflammation. So for example, in the gut, where there's a really large microbial community, when there is this dysbiosis or imbalance, the gut lining can become more permeable or leaky in simple terms. People might have heard leaky gut. Yep. And then, that can allow more of the organisms that are problematic or their products to leak into the blood, which then can cause more inflammation in the blood. So you have a lot of not just the community imbalances, but potentially barrier breakdown that leads to a lot of organisms getting into places where they're just going to drive inflammation and problems instead of again, being part of these helpful communities. So that's a trend. And what we're trying to do with our research program is, for example, there's a lung tissue study that's being run by a team at the J. Craig Venter Institute and Cardiff University in the UK. And they'll have lung tissue samples that they'll look for SARS-CoV-2 and in all these other, immune and gene changes, but they're also going to analyze bronchial lavage fluid from the same patient. And there characterize the microbiome and all of its activity. And then they can correlate and say, in patients who might have the virus in tissue, are there changes in that upper respiratory tract microbiome? We're actually just building that into the research so that we can actually just go ahead and better understand those trends, right? Yep. And by studying this, it gives us a lot of potential then therapeutic options that we might be able to bring in down the road. Because if the virus is still there, perhaps we can bring in antivirals over time. I don't think it's going to be simple because we might need to mix and match them, use them for longer periods of time. It's sometimes harder to wear down a chronic infection. If other pathogens reactivate, there's growing companies and people working on herpes virus control right now. We can maybe bring some of that stuff in. If microbiome is imbalanced, there are a lot of companies working on microbiome therapeutics, so sort of cocktails of organisms that people might be able to take to sort of replenish the communities that might have been depleted or imbalanced by the infection. So these researching long COVID in these sort of practical ways also opens up clear sort of therapeutic potential for the findings to be able to lead to. And then last, and then I'll mention, well, should I go on with the paper? Do you have any questions thus far?
No, I mean, you're doing amazing. No, keep going, because I want to,
okay.
You brought up a lot of things I'm going to ask you about, but finish your train of thought.
Okay, I'll throw in a couple more things from a few of the other sections. And one is that clotting and coagulation are definitely issues in COVID, and now increasingly extend to long COVID. And our colleagues, Ricia Pretorius and Doug Kell, and Ricia's at Stellenbosch University in South Africa, they've actually identified in long COVID blood. And actually then I worked with them to do a similar study in MECFS, and we actually found similar clots, but these little fibrin amyloid microclots in the blood of patients with long COVID. So these little deposits that are, they're small, and they're not as big as a stroke clot, because they obviously, luckily don't kill the patient usually, but they're little tiny deposit clots. And what might happen with those clots is that when they get into the little blood vessels, the capillaries that are supposed to be feeding, for example, the peripheral nerves of the body, they might prevent blood flow there, might become difficult. And so that lack of being able to get blood, and blood contains oxygen to tissues, can result in what's called hypoxia, lower levels of oxygen in tissue, in addition to just any form of clotting being associated with inflammatory signaling and nets, neutrophil extracellular trap formation that can just actually technically sometimes just damage the blood vessels themselves, the vasculature. And if that happens, and let's say these clots, and this is another study that we're working on as part of our initiative, let's say those little clots do prevent blood flow to the peripheral nerves. One of the things that has been also documented in patients with long COVID is a high incidence of what's called small fiber neuropathy. And small fiber neuropathy, what happens is a doctor gets a little punch biopsy sample of either a little bit of tissue from the patient's thigh or ankle, and they count the peripheral nerves there. And in patients with long COVID, or people who have the neuropathy, a good number of those nerves are dead or diminished. And the question is why? And one of the hypotheses that we're pursuing in our research program is that perhaps the clots are preventing blood and oxygen from getting to those nerves, and that might be part of why they're dying off. And so you can begin to connect these trends, for example, the clotting to the neuropathy, to, the whole, and with the clots, you can begin to connect it to microbiome and the virus itself, because the same team that found the clots did an experiment where they put spike protein into blood, the SARS-CoV-2 spike protein, and the clots form. So maybe a patient still has the virus, and then they have that in tissue, and then some spike leaks into the blood, and the spike helps catalyze the formation of the little clots, and the little clots block the peripheral vessels, and then that leads to small fiber neuropathy, and now the peripheral nervous system isn't functioning well, and that adds to autonomic symptoms. So you can begin to link these trends together, and that's where it gets exciting, right? And then last is a part of the paper that talks about a core signaling pathway in the human body that Mike Van Elzeker and I are truly interested in, and we think probably is dysregulated in a decent number of infection-associated chronic conditions. And that is signaling of the vagus nerve to the dorsal brainstem, which is an area at the back of the brain. And so the vagus nerve, it's a really important nerve that innervates every major trunk organ of the human body. And when it's in that organ, it has these little chemoreceptors that can sense inflammation there. So if you have an ongoing infection, for example, a reservoir of SARS-CoV-2, or there's just an inflammatory issue, those chemoreceptors activate, and they say, inflammation, right? When they do that, they convey a pro-inflammatory signal to that area at the back of the brain called the dorsal brainstem. And if you look at our paper, we drew a diagram of these overlapping nerve bodies that are in the dorsal brainstem. And they're, nerve bodies that control many of the major symptoms documented in patients with long COVID and also in ME/CFS, that related diagnoses. So some of the top symptoms, and there's many symptoms, but some of the top symptoms that long COVID patients experience are, sickness behavior. What I mean by that is flu-like symptoms, the feeling, the malaise, the feeling you get when you have food poisoning and you're sick. That there's a nuclei in the brainstem that controls what's called the sickness behavior response that modulates those symptoms. So if the vagus nerve is conveying this pro-inflammatory signal to that area, that's going to activate and you're going to get sick, symptoms saying you're sick, you're flu-like, you have malaise through that signaling. There's also really near that a nerve body nuclei that plays a big role in controlling autonomic function. Autonomic function being, in simple terms, the ability to move from a low, like laying position to a standing position without getting dizzy, to adjust to these changes in blood pressure that the body, needs to do, right? And that signaling is tightly controlled there. And then there's even another nuclei that controls pain and nausea in that same area, right? So overall, if the vagus nerve is sensing, for example, ongoing SARS-CoV-2 reservoir in different parts of the body, or even microbiome imbalance that can also cause inflammation, or maybe both, it's conveying now these inflammatory signals and it's throwing off the delicate signaling of those nuclei that control pain, fatigue, malaise, nausea, autonomic signaling. And those are the core symptoms of long COVID. So we do think that an interesting model to think about for how some of these core symptoms develop is that patients may not, for example, let's say patients still harbor the SARS-CoV-2 virus in reservoir. It doesn't even have to be in the same location. Maybe someone still has the virus in the lung, someone in the gut, but the vagus nerve innervates those sites. So there can be sort of a common, effect on the dorsal brainstem that can happen in those different patients. And let's say Epstein-Barr virus reactivates in one patient in addition to SARS-CoV-2. That will also be sensed by vagus nerve. So it helps to a point. We think that that's circuitry, as we call it, maybe a common circuitry that's impacted in different patients with long COVID, even if the exact infections in their cases, the reactivated pathogens are, and the location of the infection is somewhat different, that can help explain, like I said, some of those common core symptoms.
It's needless to say, it's an extremely comprehensive paper, and I appreciate Amy walking through it with us like that. And really, you got a lot of things to work on. It clearly, as you were saying, the rainforest, the jungle that is long COVID has many tentacles for you to go after and explore. I was just wondering, because you mentioned the gut microbiome, and it's interesting. Just like a month after you published this paper, you published another paper. It was in Nature Microbiology. This one was entitled "Metagenomic Compendium of 189,680 DNA Viruses from the Human Gut Microbiome." And here you go into well, you go into a lot of different things here, but you're mentioning the bacteriophages before, which, infect bacteria that they can also be useful for us in certain ways. And it just got me thinking. I know, I guess it's tough to look for silver linings in some of this stuff, but I've done some episodes before talking about sort of the symbiotic, sort of the beneficial virome. Talk to folks like Jack Stapleton, University of Iowa, and so this hepatitis G that makes us, it less, protects us against things like herpes and HIV and so forth. Any silver? I mean, I know it's kind of early because you just, this has only been going on, and I think it's amazing that you not only set this whole organization up but raised money for it in such a short period of time. But any interesting benefits? I mean, I'm not saying benefits along COVID, but any positive biology here that you may be able to pull out of this? I mean, it might be a silly question, but just thought I'd ask.
No, I mean, I think actually that while it sounds, first of all, it's concerning that this is happening with these cases. It's actually exciting to have clear leads on what to study and what to do, because if we're able to come in with what we call a hypothesis-driven approach and say, we're not coming into this study just hoping we find something in your blood or something.
Right.
We actually have a pretty good idea of what we think might be happening. And so we're able to design a study very specifically to try to answer that question really intelligently. We might actually get a good answer. And then when you actually know what you're looking for, this is the biggest part in these diseases. When you know, and especially if what you identified might be root cause, actually sort of at that level, then I think that that's a good thing. What you're doing is you're taking something that was otherwise mysterious and almost just, to me, it's a lot scarier and more disconcerting to not know what's wrong with you than to actually have a more concrete idea of what the problem is, even if you don't have a perfect solution. But knowing the problem, then, there are a lot of biotech companies, there are a lot of intelligent people out there, and being able to document the problem really clearly, I do think that can actually create a lot of robust drug and other development. And then there's actually a lot of energy in that space to do this. So I think that overall, actually, I hope that there can be a lot of positive outcomes of this, where we actually, there's a lot more teams that are coming into long COVID. So, we've had some scattered teams on some of these other infection-associated conditions before this, is the honest truth. And so many labs started studying COVID because of the pandemic and started developing really good methods for SARS-CoV-2 identification and related trends, that now those teams are able to move into long COVID really rapidly and quickly and iterate what they're doing. So we have this whole new growing talent coming into the community, which is amazing. And so with so many more minds on this as well, we're going to be able to move and find things. And I think that our understanding of what happens in long COVID will have great relevance to related infection-associated chronic conditions. And in fact, we might even be able, one of our goals is to take some of the studies in long COVID that we're doing, and then after we finish in long COVID, do a similar study in MECFS or even chronic Lyme disease, or, so we can, now that we have the infrastructure for the study, now that we have everything, the teams of this, we can then start to adapt to different viruses, different pathogens. So it actually really we're setting up some of these really excellent pipelines that I think will actually bring, again, really good research into some of these other conditions that are related, but actually have been understudied and honestly underfunded. But yeah, so I actually see a lot of positive potential outcomes there. It will take a lot of work, but I do see positivity in the future. And with the phage, so the other study you discussed, though, that's a whole other side, fascination of mine. That's led largely by a team, Nikos Kripke, that's at the Berkeley Joint Genome Institute. And that team has developed these incredible technologies for identifying bacteriophage, those viruses I mentioned that, when you get into this kind of research, this is just exploratory. This is a little bit like exploring outer space, except it's the human body, right? So in fact, phage, these bacteriophages, the viruses that infect bacteria in the human body, are still referred to by some teams as dark matter, because there seem to be, I think, a current estimate from a researcher at Stanford, quadrillions of these bacteriophages that traffic human blood and tissue at any given time. And we've only really identified and classified and named a small fraction of them. So we have these, unnamed, uncharacterized phage just in and on us, right? So it's as a new frontier. And that's what that paper is doing, is Nikos and team spent a long time developing technology to identify both known but also novel bacteriophage from samples that they were doing in the ocean, other environments. And then I did slightly help convince them to turn those technologies on the human body. I was like, look, you can do this. You can find known and novel bacteriophage. Let's turn that under the human gut. And yeah, when they've done that, that's a paper. It's just, that's what the paper is. It's just coming up and saying, we found all these phage. No one's ever documented them before. Let's go ahead and, give them numbers. Let's go ahead and put them in databases. And then, the next round, we're working on another study now. We improved the tools. So the next round is going to find more, but that's what it is. It's sort of mapping what is inside us. And we don't know, we don't know exactly what all these phage are doing. It is possible, that we obviously, people are some people can be completely healthy. They can be an Olympic athlete and they have all these phage. They definitely are not necessarily detrimental, but there may be health conditions in which a shift in the phage, or the bacteria that they infect might be part of it. Maybe exactly when, the bacterial microbiome gets thrown off, the phage do too, but then there might be also therapeutic approaches. So we can think more then, if we know more about the phage, right, we can think about, well, how do we regulate them back? Yeah. And with phage too, as you probably know, you can maybe iterate a phage into what's called phage therapy, where yeah, since you have these viruses that kill bacteria, sort of that's what they're doing. They're just infecting and killing bacteria as part of this ecosystem. Let's say a person's infected with a really bad bacterial pathogen that antibiotics are not working for. This happens, and they can't be treated otherwise. Maybe we can find a phage that infects that bacterial organism and can kill it. And so that's an ongoing area of research as well. And that's called phage therapy, potentially using a bacteriophage as a therapy to kill a bacterial organism that's resistant to other forms of treatment. So there's a lot of potential, then translational potential, to better understand the phage in the human body.
Yeah, it's that's one of the areas that I'm personally fascinated by. About 2 years ago now, I did a show with Chip Schooley at UC San Diego, and here you have, as you say, here's evolutionarily perfected viruses that have been around a pretty damn long time and pretty good at killing microbes. We have a lot. I mean, I think, correct me if I'm wrong, I think phage, didn't they come up with that concept before we learned about penicillin? I mean, it was a long time ago. And yet it's still, I think there's like 2 countries in the world where we actually even practice it. It gets me thinking of, back to your intro and talking about sort of your own self-experimentation. You weren't, when you were dealing with your own issue with Epstein-Barr and so forth, you weren't looking at sort of single magic bullets. You were trying antibiotic cocktails, and you mentioned some natural antivirals, things like that. Talk a little bit about that, because I think that's something that, the whole area of antimicrobial resistance has been really hot in recent years. I think I don't know what the last antibiotic we developed was in the 1980s at some point. So we're kind of struggling with that. And it seems like for something like this, it's not going to be a single magic bullet. You're going to have to come up with interesting cocktails. You're going to have to mix and match. You're going to have to explore the potential in nature. Talk a little bit about sort of like the ideal. I know you can't, this is only beginning. What does the ideal intervention to you look like for something like this? I mean, what types of things are you going to try in terms of repurposing, in terms of natural products? Take us on a little journey on that front.
For something like long COVID?
Yeah, any of these these conditions where you got multiple things going on.
Yeah. Well, you're absolutely right that there's unlikely to be the cure. You'll hear that sometimes, like, team finds cure. No, I mean, no. We know from cancer and other conditions where the therapeutics have actually advanced more, recently, that no, we're talking cocktails of drugs, different, certain drugs that modulate the immune response while bringing in other drugs that might affect organisms. It's going to be a combination of therapeutics, and the therapeutics may actually differ from patient to patient. I mean, it's really time that we enter a true era of personalized medicine, where what we're going to have to do is say, for example, in long COVID, like, okay, do you still harbor the SARS-CoV-2 virus? Okay, if so, where? What other viruses or pathogens or bacterial pathogens may be involved in your case? Yep. What else is going on with the immune response? How much clotting do you, it's a personalized profile of how that patient got to their certain set of symptoms. First of all, we need that. And then from there, treatment is almost tailored to the patient based on that understanding. But of course, within that, there's common trends. I mean, with the viruses themselves, let's say SARS-CoV-2 or Epstein-Barr virus, it is far time. And I think that this is leading, there's been some shift to really robustly make it a large human effort to develop more antivirals. We are, when you say we have very few antimicrobials, we also have a really limited number of antivirals, considering the number of viruses out there and how much we know they can kind of kill us, and how we're in a pandemic. We should probably be putting a large amount of our science and biotech talent into antivirals. And actually, the COVID pandemic has at least started some antiviral development. I mean, it's interesting to see, Paxlovid be developed, molnupiravir, for however we think these antivirals work. We definitely need more data on them, is the honest truth. Remdesivir, at least it's been great to see companies actually developing new antivirals. It's a good step in the right direction. I do think that we need to better understand, though, we first need more data on how they are working in acute COVID. It's not even totally well understood. But as we move into long COVID, let's say someone still harbors the SARS-CoV-2 virus later. The honest truth is that once a pathogen is sort of in a more latent or persistent state, depending on what you want to call it, it might be a little bit more embedded, and you might need longer courses. It might not be as simple to eradicate as when it's in what's called the acute state, where it's still just, sort of, there's more virus, but it's still in the blood. It's still in, it's not ensconced as much in a chronic state where it might be a little bit more adapted to the body, right? So in that context, we also might need to think through longer courses of antivirals, pulsed antivirals. For example, what if the patient takes 2 antivirals at the same time, and one has a mechanism that inhibits viral replication and another has a mechanism that inhibits entry? We're combining different antivirals with different mechanisms of action. That's going to make it extra hard for the virus to survive in the face of them, right? So I think there's going to need to be some ingenuity in the way antivirals are used and trialed in clinical trials. And then beyond that, there's a couple, companies that I know of that are trying now to develop, Epstein-Barr virus antivirals. Yes, please. I mean, we really, we really do need that to happen. We need to take these viruses seriously. I think, there's been the last decades or so in science have focused a lot on autoimmunity, and we've developed a good number of immunosuppressive drugs. So drugs that hold down the immune system, that sort of just palliate and take down inflammation. But the more that we understand, for example, like in a multiple sclerosis, that viruses may actually be at the heart of what we're calling autoimmunity, the more those immunosuppressive drugs don't seem like ideal options for a lot of patients, because what can happen is you are knocking down with those drugs the inflammation that might be associated with an infection or with a problem. But if there's a pathogen at the root of that, it's actually almost a little easier for it to just hang around, because the immune system is less robust. And now when the patient's on immunosuppressive drugs, they're somewhat more susceptible to other pathogens and to the next infection. It's not a great model for getting people totally better. So I do think we have to continue to move as often as we can away from blunt immunosuppression, just holding down the immune system, which can often just be largely palliative. It can help manage symptoms, but it doesn't really get to the root of the problem, and into immunomodulation. And what I mean by that is you're actually trying to understand how the immune system is dysregulated, either by a pathogen or just a process. And then you're activating or modulating certain components intelligently to be able to restore that situation. So in cancer, there are now T-cell immunotherapies, and those are pretty amazing. What they can do is they'll take a patient's tumor, they'll, understand the sort of personalized mutations in that tumor, and then they engineer that patient's T-cells to go after the tumor. And that it actually leads to what's sometimes called immunopathology, or immune reconstitution syndrome, where as the T cells that are now re-energized attack the tumor to break it up, there's actually an increase in symptoms that the patient experiences, because the battle between the T cells and the tumor and sort of the debris causes a rise in symptoms. But if the patient's able to be managed and through that process, their eventual outcome for their cancer survival is much greater. So the same is very possibly likely true for something like long COVID. We know that SARS-CoV-2 is holding down interferon signaling. We know that it's muting T-cell responses, and a good number of cases are modulating T-cell activity as well. So perhaps we can also bring in a T-cell therapeutic or T-cell vaccine that brings T-cell activity back, so the T-cells can better target the virus. So we can use maybe the immune system as well, and not just the antivirals we make, but our own immune systems, and give them the strength to better target a pathogen or whatever other issue might be going on. And then if you combine that, like I said, with with therapies that also try to restore the balance of the human microbiome, I always consider that to be helpful. I always think that if someone's chronically ill enough, it's probably the microbiome probably isn't in the best shape. And with microbiome, what you can do is there's a book called the Gut Health Protocol, and I've done that protocol. You can order the supplements from your home. Basically, it's a simple concept. You take some supplements, and sometimes people will do antibiotics, but you don't even have to, depending on the supplements, to sort of kill what might be the overgrowth of fungi or bad bacteria. And then you try to replenish the gut with probiotics, fermented foods, and better bacteria to try to bring those organisms in there to try to take hold more, right? And you can do these kinds of protocols. And I think a good number of people, if they really do these, I'm not saying it cures them, but that they benefit. And there's also certain supplements that can potentially help with the gut lining. One of the studies we're also supporting as part of our long COVID initiative is a study with a team at Harvard that actually can measure a marker of intestinal permeability called zonulin. And then they have a therapeutic that it seems and zonulin is sort of upregulated when the gut becomes leaky or permeable. And they have a therapeutic that might be able to repair that, right? So like if you improve your microbiome balance, and then you also can maybe take something that sort of heals the lining of the gut so that it's no longer leaky, and you're combining that with antivirals or other medications to target the major pathogens that might be involved in a case, and you're strengthening or supporting the immune system so that it can better function. If you're doing that over time, then it seems like there's recovery, improvement possibility in doing that.
I like the way you think about these interventions. Very elegant, and the combinatorial concepts are that speaks to me.
Yeah.
And so that's when we talk about the silver magic bullet that sort of permeated this industry for way too long. It's very refreshing to hear you talk about this. Amy, what you raised the $15 million was extremely impressive for a sort of a new condition that's only been around a short period of time. You look like you have a goal to boost up to about $100 million. Just talk a little bit about what's going on that front, and a little bit of what was the fundraising process like for this? How long did it take you? I mean, you have some impressive backers here, Vitalik Buterin, Patrick Sinshang. Talk a little bit about that whole part of the story, because I think it's pretty impressive as well.
Sure. I mean, Vitalik Buterin was amazing as a donor because, what happened is he created a fund for high impact COVID research. He actually took the pandemic seriously and decided that maybe he should actually chip in and try to help a little bit with the pandemic situation, which I appreciate. And so he got a good team together of just interested scientists and helpers who took a realistic look at the space. They got on Twitter, they talked to patients, they talked to people, and they figured out who was sort of really just moving things forward and actually, making discoveries and moving. And they funded some things that are just awesome and basic. For example, better filtration devices. A lot of what they're working on is they're trying to fund products or things that can be used by people all over the world, which is a really admirable goal. So what they funded, for example, are just affordable filtration devices that someone might be able to build even in their house in Africa, right? Not just for people who are wealthy. Also other, things along those lines for just being able to sort of do some basic containment and to help people across the globe not get COVID. But they definitely noticed, and it's not hard, long COVID, right? Which is just one of the more terrible consequences of COVID. People who are technically chronically ill for life after this infection. And they took that really seriously, and they decided to dedicate a good amount of their money to long COVID. And I met them first because they agreed to fund an intestinal tissue project in long COVID that is really one of the most important studies happening right now. if the virus, the intestine, the tissue in the intestine is a known reservoir site for viruses, and there's already the team that's on that project has already found the virus in intestinal tissue after acute illness. So now that's happening, that study in a cohort of long COVID patients. And if virus is identified there, like we said, that's a clear therapeutic target. Now they're funding some of the most just direct, impactful studies with some of the top teams in the space. And so what we did is we took the other researchers that we knew in the space that were just had some of the most experience and were ready to move on top projects. And we created a roadmap that we showed to them for how we could just make many more impactful long COVID studies move forward with funding. And they were willing to give us the chance with the donation, and it was really amazing.
Awesome. That's really awesome. One other thing while I have Amy, because I became familiar with you a couple of years ago, and I saw you on some different platforms and shows talking in general about sort of the microbiome and human health. You published a lot on vitamin D. I know you got a lot going on with the long COVID work now, but anything else exciting happening in 24 hours a day? But anything else going on some of these other fronts that we associate you with in terms of just human health and wellness?
I mean, mostly focused on long COVID these days with the goal to move into ME/CFS and definitely chronic Lyme disease. One thing I will emphasize is that the tick-borne pathogens are really dramatically understudied and appreciated in people with chronic disease. So it's a big point of ours to help bring in the top technologies and even improved technologies for identification of tick or vector-borne pathogens as part of what we're doing as well. We are not leaving them out by any means, and that's an important goal. And I'll just say that honestly, that I'm just a huge organism nerd still. I always have been. I'm just a huge infection and organism nerd. So one of the favorite things that we're working on is a tissue analysis pipeline with a team at the J. Craig Venner Institute and also the Berkeley Joint Genome Institute, where our goal is to see if we can succeed in creating a pipeline where we could take a tissue sample and via the use of different overlapping advanced technologies, be able to identify bacteria in that sample, fungi in that sample, archaea in that sample, and then DNA viruses, RNA viruses, all bacteriophage, and then known or novel DNA viruses and bacteriophage in that sample. And we'll see, we actually have a pretty amazing pipeline that we got a large grant for to work on. And there are teams that can find bacteria in a sample and teams that can find viruses in a sample, but to our knowledge, this would be the first that could try to do it all. And so I am a big nerd when it comes to that one, just trying to push the boundaries. What I most want, I want to know what organisms are in me. I want to know what pathogens I have, and I want everyone else to be able to have that information. That information is so key to understanding both health and a wide range of chronic conditions. It is key to even understanding aging and longevity. People, we now know one of the most interesting things, and another team we collaborate with who has a member on our scientific advisory board, they've actually found that in Alzheimer's, the plaque, what we call plaque, actually, and this is Rudy Tanzi's team at Harvard, actually seems to act as an antimicrobial peptide, which is part of the immune system that forms in response to pathogens in brain tissue. So there's a pathogen there, and then the antimicrobial peptide forms around it, digested, and now, yes, it is a plaque, but the reason it formed is because it entrapped a pathogen. The implications of that research, which is growing and more teams are following up on this, is huge. In Alzheimer's, we've just been trying to remove the plaque. All these drugs keep failing. The rates of Alzheimer's going. What if we actually target the pathogens or parts of there's so much potential to just really change our trajectory on diseases of the aging and other conditions, Parkinson's, through related research on infection. There's growing infectious components to so many of these diseases. And by doing that, and by studying this topic, infection and organisms, then what it really means is we have huge potential to just lower the burden of chronic disease. So that is a goal.
Outstanding.
Just keep doing that.
Really outstanding work. I mean, it's so impressive, the portfolio that you put together here, all these different programs, and just really rooting you on this one and all of it, because it, as you said, the microbiome, the virome, it just touches on pretty much everything. And yeah, I mean, for everybody that's going to be listening to this particular episode of our show across the different podcast networks, or watching on our YouTube channel, again, you've been listening to Dr. Amy Proal, President and Chief Executive Officer of PolyBio Research Foundation, also Chief Scientific Officer of the Long COVID Research Initiative. Amy, I want to thank you for taking the time out of your schedule to come talk to us and educate us for a little while on these topics. Obviously, thank you for everything you're doing there to move this area of science forward. And as we say on our show, thanks for helping to create a better tomorrow for so many people via the work you're doing. It's really so impressive.
Thank you so much. Thanks for having me on, and it's fun to talk. I love talking about these topics, so no problem.
There you go.